You guys hear me well?
Yep.
Yeah, okay. Thank you so much, Thomas, for the kind introduction, and also to the organizers for inviting me to this version of the Pacific Northwest Numerical Analysis Seminar. It's a great honor actually to give a talk alongside very renowned researchers in the Pacific Northwest area. Um, so today, as Thomas was saying, I will be talking about some aspects on skeletal muscle modeling. So this is part of what what I was doing at SFU for my PhD. Um, and now I'm actually very happy because there are some connections with what I'm starting doing with the group at the University of Saskatchewan. So I do see, I'll show at the end that there is some future between the two projects that I have in my back pocket. Um, so, so in my first slide, I have something that is not usual. So usually right underneath the name of the conference, I would put the university where it's being held. But this year I just put the universities where the three organizers are from, because I didn't want to put Zoom on the slide. Okay. Um, so So I will start talking about the team that I was working with back at SFU. Some of them were at SFU, some of others were not. First, my two supervisors were Neil Manigam in mathematics, that maybe many of you know. My co-supervisor was in biomedical and physiology, uh, James Wakeling. Um, so this is where my kind of this mix of math and biomedical areas came from within my PhD, which was very useful at the end because I ended up learning a lot of the non-mathematical points or details of the model. My two other collaborators were David Ryan, also used to be a PhD student who is now at U California Irvine, I think, as a postdoc. PhD student Stephanie Ross, who's still there. She is finishing very soon. Postdoc fellow Bart Borstoli, who is in a which is a Neuroscience Research Australia, who was visiting us last year. We actually had a very nice time while he was there. We learned a lot from his 3D MRI scans that he did. I'll talk about that later as well. Ryan Kahn, who started with us as a UBC student in physics, and now he's a master's student in mathematics, who is currently working on the model as well. He's extended it to some other uses. And the last one is Cassidy Tam, who's she started as a undergrad student from McGill, and now she's at SFU in the master's program, and she's working on different aspects of this project. So in particular here, I have a picture of something that we did when Bart was visiting us. So essentially we took this MRI data, 3D data, and we meshed it. And then we tried our model. Basically, was the first picture that we got out of the code with the MRI data in. So I was very happy to see this. And then I'll tell you later, but we compared it with some real data, and then we saw something very close, very similar to what they measured in real life. So I'll start with the goals of this project. So when I started, the main ideas were to design a numerical framework in which we could implement many different experiments that people do in the lab that we could do on the computer, okay? So one of them, for example, is to use MRI data within the code, right? And then run several experiments on that. Or sometimes do lengthening of muscle tissue and see what the properties are or what the forces are where you do that. The other aspect was how to develop semi-implicit time stepping, and I'll get into more details about that later, and why we wanted to do a semi-implicit and not like an implicit, which would be something that many would try first because there is no much knowledge on on the stability of this problem. We wanted to study the effects of mechanical properties of different tissues. So muscles are units that are composed of many, many different tissues. And so we wanted to see if we added one more, what happened to the output, if we were getting closer to what we were supposed to see or not, and things like that. We wanted to study some dimensionality effects, mainly because most of the current models are based on 1D simulations of muscles. So usually researchers think of muscles as a 1D object. So like you can think about it as a 1D nonlinear spring, and then they compute things and they arrive at conclusions from that model. We wanted to study how the volume changes, of the volume of the muscle changes during the deformation of the tissues as well. Many models actually don't consider that, so they are incompressible in that sense. And the last thing that we've been trying last year was to explore and trying to understand how the energy flows within the system. So once you give like a right-hand side to the model, what happens to the energy, how the energy flows within all the components that we have in the current model. Okay. And there are many other goals that I didn't want to put too much text on my slide and a picture, so I changed that. So essentially, all these goals also come with some challenges, of course, right? One of them is, as usual, probably in the biological systems, is that the complexity of what you're trying to model is pretty high. And when I say complexity, it means that the geometry is very complex. The mix of different materials is very complex too, right? Getting 3D images of muscles that are like attached to your body when you're alive or when animals are alive is not easy. And so many of the measurements that you find out there are based on dead muscles. So like I always, well, when we used to meet in person, I would tell this joke that like you could go to the superstore or the supermarket, right, and get a piece of meat and then try to test it at home and see how long you get, how many data points you can get with that. Now, I don't see people laughing because I just see you guys very tiny, so I don't tell the joke again anymore. So again, one of the main features of muscles is that they are based on fibers. Okay, so a muscle, like imagine, I think I'm gonna go back to the chicken now. I'm gonna go like to the chicken when you cook. Like imagine you put a piece. I'm sorry about the vegetarians. Maybe that's not an example for you guys. I was a vegetarian sometime, but so imagine you put a piece of chicken breast and you cook it, and then you can actually pull some of the fibers, right? Like you would see some fibers when you kind of like tear apart. So those are actually kind of the main structure in muscle. Okay, so they are fibers, and these fibers produce forces in some way. Okay, so in our model, we wanted to capture this, and actually one of the main and the most basic unit in our model is that fiber, all right? That produces forces, and it's got elastic properties as well. Obviously, after you cook it, there's not much elasticity left, right? I do see Grady laughing, so I'm gonna keep telling jokes, bro. So as I said, the goals, right? Like to get there are many challenges on the way. One of them is the architecture, right? The geometry. There are different materials, as I said before, and there are fibers. And because we want to consider these fibers in the model, you can think of these fibers as a curve in 3D, maybe with volume, right? It's like a cylinder in 3D. But at every point, you have different orientation of this fiber, right? It's not just a straight line. Sometimes it is a straight line, but others it's not. So we assign this orientation A naught to each point where we compute the solution along this fiber, right? And it could go like this and then go down and up. It could differ from point to point. We have different materials, as I said. So currently we have the capability of having muscle tissue. with its properties, tendon with its properties, something called aponeurosis, which is it's something I had no idea about before I started this project. Of course, for me, muscle was well, the whole muscle was just the muscle and the tendon. But this aponeurosis is like a very thin sheet that covers the muscle and it extends from the tendon. So tendon is usually a very stiff material that connects the bone to the muscle. So the bone is never connected directly to the bone, but there is a tendon in the way, in the middle. Okay, so there is a tendon, the muscle with very different properties, and then on top of the muscle you can think of this tendon like an extension of the tendon, but it's a very thin layer of it, and it covers one side of the muscle. But even though it's an extension of the tendon, the properties, because of geometry and it's got fibers in it as well, the orientation of these fibers make the material different, behave differently. Okay, so we wanted to add that as well, because like we would see this in the simulations once we were just doing a muscle-based simulation, no tendon, no aponeurosis. We would see that um, it was not as stable as we thought it would be. Like we were in to get to the numbers that we were seeing from some experimental data, we would have to do something. And then when we put this aponeurosis on top, we saw that some things were kind of coming together. Okay, so it does something in the model. And this is actually something that we haven't seen before. So this was something new we wanted to try, and it's working very well for us so far. The last one is a very important piece of the model, actually, which is the base material. So I don't know if you can see my screen here, but here I have a little sketch of a muscle. So our smaller unit will be called fiber or muscle fiber. And you can see that there's still things inside this muscle fiber, okay? So we don't go into that so much into those details anymore. There is a group actually at University of Washington, they do simulation at the smaller level, like a microscopic level with cells and things like that. But we don't, we look at the problem from the microscopic point of view. So muscle fiber will be the smallest unit, and you can see that there are other materials around the fibers. They are not alone, of course, because if you put fibers all together and then you don't glue them together, right, they're gonna fall, right? So there is something that keeps these fibers together, and this thing is actually made from many different materials. So what we are doing currently in the model is to call all this material that is surrounding these fibers, we call it base material, okay? And why is that? It's because our model currently is based on data that we find on available online, right? So each piece, like we would have some tests on some material, and then we would use that to fit the model. And unfortunately, it's really, really difficult to test the base material in the lab without having the fibers. So far, we haven't been able to separate all the list of things that I have here of materials too much because of lack of data, you would say. And also because that brings more complexity into the model, right? We shouldn't just blame it on the experimentalist, but it's also because that would bring more complexity to the algorithm, and it would become more expensive, right? The other challenge is that, as you may think, like when you're working out or you're lifting, you're using your muscles, you can see that your muscles deform, right? Which is quite far from what we are used to and analyze, which is like linear elasticity, right? In there, you see very small changes, right, in the elastic body, but here you can see the deformation. So this problem is a large deformation problem, and it's nonlinear, right? Because once we put together many fibers, right, with the base material and the fibers, as we will see, the material, the whole unit will behave in a different way when you are along the fiber direction compared to the transverse direction, all right? As I said, we knew, actually, there are a few papers where they study the compressibility of muscle tissues. Actually, I've seen a range from 4%, 2% of changes in volume to 20%, right? The 20% is a bit different. I can, maybe if somebody has a question about it, I can comment on that quickly. But essentially, the volume changes during the deformation of the muscles. And there is a very particular property related to within the muscle fibers. These are that, even imagine a linear system, or a nonlinear system, actually, it can't be a linear system, but the right-hand side and the boundary conditions are all 0. So you would hope for the nonlinear system to have a 0 solution for that. Well, this guy here is not, actually. So these active forces I'm referring to, which is the red curve here, are actually contributions from electrical impulses from your brain that actually make your fibers shorten from its initial length. And that would generate a force. So there you have something that is common, is considered an internal process, and it contributes to a solution, even though you have nothing, like no external contributions to the system, all right? Okay, so how did we base our model, our 3D model? So there is a very famous model called the hill-type muscle model, which is essentially looking like this. It's a non-linear spring, all right? So this spring here is called the passive elastic element. So essentially that encloses all the information about the elastic response of the fibers of the muscle, of the whole muscle. So these vectors here are representing a whole muscle now, not a fiber. And this CE fast and CE slow, they refer to these electrical impulses that come from the brain down to the muscle, and they activate the fibers and make them produce forces, all right? So this model was introduced in the 30s, a long time ago, almost 100 years ago, but it's still being used today, okay? And it's because roughly it can approximate, not very well, but it can approximate forces that muscles produce as a whole. But it doesn't know anything about the structure, what's happening inside. So things like studying the energy flow of within a muscle can't be done with this kind of models, for example, right? And so here I have a few references. The last one is 2018, where this model was used in series and in parallel to see how those things, how the different configurations would change the output of the model. And then we wanted to use that in our 3D model. So essentially what we're using currently is a version of this locally. So there is a hill-type muscle model, a version of this model for muscle, for fibers, and for something called sarcomeres, which are units inside the fibers. But there's also a version of this model for fibers. We say, okay, maybe we can take this model and put it locally in parallel, right? Or in series first, and then by putting fibers next to each other in parallel as well. And that's how we based our model, all right? And this is not the first time that people do this. There are a few papers where they do something similar. And our work is actually based on Rahami's work, where they came up with this idea of taking this hill-type muscle model to 3D, okay? So there are many interactions here now from here to here that make it a bit different. Okay. So we have a few assumptions. So we assume that as in the real life, fibers run throughout the muscle unit. So they go from point A to point B inside the muscle. Different materials. So we have muscle, tendon, aponeurosis, fat as well. We have it in the model currently, and the basic material. One of the students, Ryan Kono, he's actually working on extracellular matrix material. So it's a bit different. So now we take kind of a combination of the base material and this extracellular material. We assume it's nearly incompressible. Each of the tissues, the muscle, tendon, and aponeurosis have fibers. So the muscle fibers are a bit different from the tendon and aponeurosis fibers. They are, in a tendon and aponeurosis, they are mainly composed by collagen, okay, which give them different elastic materials. And they can't be activated as the muscle fibers, right? We assume that the base material behave as an isotropic nonlinear material. So it means that if you have a cube of it and you press it from here or from here with the same boundary conditions, right, on the other parts, you should see the same result. And overall, putting all these things together, muscles actually behave as an anisotropic material, right? This is what I said before. So if you're along the fiber, you will get one response, but then orthogonal to the fiber, right, you will get a different response, right? Here is what I said before. So these sarcomeres are actually inside this myofibril, okay? But we don't consider this currently in the model. We just go down all the way to muscle fibers, all right? This is our smallest unit. Okay, so how to formulate this problem? So if we go back to continuum mechanics, probably in undergrad or grad school, a general kind of solid mechanic problem looks like this, something like the first two equations. Okay, so there is a mass conservation and then you have the balance of forces, right? But in our model, we based our formulation on work done by Simo in the 80s and 90s, early 90s, okay? Where he proposed to add this P as a Lagrange multiplier, which ends up to be the pressure, and also to work with orientations, with fiber-based materials, right? So once you formulate your problem, you end up with these two extra equations. So the pressure is related to a part of the energy, which we call vol, okay? And it depends on this D, which is called the dilation. So the dilation is the amount of volume changes that you're seeing over time, right? Locally. So we compute that locally, all right? And of course, to close the system, we need some initial conditions here, right? Okay. So, so there are two terms actually before I move on. There are two terms here in the potential energy, the phi iso and the phi vol. And we do this split because of changes in volume, right? So we want to quantify how much the volume is changing. And this nicely ties up with the definition of the pressure. And then this phi iso will have all the information from the elastic properties of each material, okay? So this is what we do. So we take this phi iso and we split it for each material, for each tissue that we have in the model. We split it as a base material. So the base material in tendon is actually different from muscle as well, okay? And the tissue itself, and this tissue is actually referred to the fibers, right? So when your material has no fiber, like in fat, you would only have this base, okay? So base is always there, right? So we have this split, and the base material, it looks like this. So it's based on Yeo's contribution in early 90s. He actually developed a few models for nonlinear isotropic materials, actually in particular rubber. And one of the models is this one, okay? And we added this constant in the front so that we would have the freedom to play around with the stiffness of the muscles. And why is that? Is because there are many diseases and conditions where your muscle actually get a little bit stiffer than usual, right? Or more compliant too. This sigma naught is actually called the the maximum isometric stress that I'll be talking about it later, okay? So this energy here is based on the first invariant. I don't want to go into details with the definition, but it's based on the first invariant of these two tensors. It's either works. Okay. Within the tissue or the fibers, we have different kinds, of course. And for the muscle, which is the most important probably, we have two parts, as I said before. So we have forces or energy that are caused by shortening or lengthening your fibers, so the elastic response of it, but we also have the active response to that, all right? And the active response encodes the information that comes from your brain telling your muscle to deform, all right? And so the active force or active stress here, which is this whole thing, A times sigma L sigma V, looks like this when the sigma V is 1, okay? So when the sigma V refers to stresses that develop due to velocity. So this is kind of like a mix with fluids here now that we're talking about velocity or stresses due to velocity, right? And so when this is 1, this force here, this stress here, sigma L, which is called force length or stress length, looks like this thing here, all right? So at 1, when you, so this is called the 1, stretch 1 is called the optimum length. It's the place where you get the highest force from the muscle, right? And this function here is very important, actually. This is called the activation function that we use. And it depends locally in space and also on time. And you may think that once you get your electrical impulse from the brain, from the neurons, right? There is a little delay in time until your muscle gets activated. So when it starts to deform due to that electrical impulse. Okay, so this tries to mimic that, all right? So you have these two parts here. As I said, when this is 1, which is actually, there is a very important type of contraction or deformation in biomechanics, which is called isometric contraction. This stress here actually becomes 1, okay? So you only have stresses due to lambda, which is the stretch, okay? So as you lengthen or shorten, you will get a different stress. The velocity doesn't count in that setting, right? For tendon and aponeurosis, the active part is 0, okay? So when your brain sends the signal, the tendon doesn't know about it, same as the aponeurosis. They just respond to the deformation within the muscle, all right? Once they see the energy in there, okay? Oh, I didn't comment on that. So the force velocity, the velocity here, or this term is actually called force velocity or stress velocity, looks like this. So as you go, so your velocity becomes larger, this becomes more and more flat. There's like an asymptotics there. And then as your velocity becomes more negative, okay, it looks like this. So it goes to 0 actually. When you're at the smallest velocity, you should see a 0 stress, okay? And it was, at the beginning, it was very strange to me that it looked like this on this side, that it was asymptotic. I thought that was just something that mathematicians talk about, right? But actually, what happens is that usually they don't have much data past 0.5. Okay. So in the experiments, it's very difficult to get to this region. So they essentially, it's like almost like an extrapolation, right? Like the curve that they are fitting there doesn't have much data, right? Okay. So to approximate these things in 3D, what we wanted to do and why we wanted to do, I'll tell you in a bit, was something like this. So we wanted to do implicit in these two equations and then semi-implicit in the velocity here, okay? And then the updates are as usual for the boundary conditions. And the reason for that was that before starting to work on the dynamic problem, we already had a good code for the isometric contraction, where the displacement U, this guy here, the pressure and the dilation were already an unknown. So we wanted to take advantage of that, right? And so if you do something like this, the next thing is that the velocity doesn't need to be in the linear system at the end. Because this system is not linear, which means that you have to apply a Newton iteration on top of that, right? Or some other nonlinear iteration. Okay, and then so we turn it actually into like a second order, or well, a single system in the UN plus one, Pn plus one, and the n plus one, right? So we essentially what we do, we only update the velocity. We don't compute the velocity at the end, right? So as I said before, one of the important examples are isometric contractions. And in the field of biomechanics, when they refer to isometric contractions, this is actually, I don't know if I have time for that, but it's a very funny story that we've been discussing. I don't want to say fighting because I want to look professional, right? But we've been discussing for a long time what it means to have an isometric contraction or fixed end contraction or blah, blah, blah. Anyways, after all of those discussions, hours and hours, what I can tell you from my point of view is that if you have a muscle that is usually clamped at one end, at one bone on one end, and then at another bone at the other end, an isometric contraction would be that if you just like put a weight on your hand and then your muscles, if you don't want your hand to move, your muscle has to contract to do that because you're putting some weight here. So essentially what I'm trying to say is that you're changing either this T, or a right-hand side, okay? So essentially what I'm telling you is that these two ends don't move at all right? This is what it means by no strain rate, okay? Why? Because the strain rate comes from the idea of the heel-type muscle model, and for them, muscles are just a 1D object, right? So it's a slightly different idea, right? So essentially that's an isometric contraction. So again, for example, for this muscle here, which is a 3D subsample of this MRI scan here, what we did was to clamp this side, okay, or corresponding to this upper side, and then clamp like a few faces here at the bottom. So this muscle here represents your gastroc on the leg, on your calf, okay? So this side here, the smaller side, is actually attached to your Achilles, and so it's the Achilles tendon here. And this other side is actually attached to the back of your knee, okay? And so we wanted to run a few simulations with that and see how it went, okay? We compared with a 10% contraction actually, that I'll show you later, and you can see how close we get with that. Okay, and so the other assumption is that there is no body forces, which means essentially that there's no, like gravity doesn't affect the deformations, which you can have your own impressions, right? Like when I heard about this was like, whoa. And it's we use a quasi-static regime for that, right? Oh, what did I do? Okay, there. Right, so quasi-static lets us to compute something, right, based on the previous time step, right, and then we can continue computing things so that in one shot we can have many frames, right? All right, so a few results here. So this is a paper that I was working on with the group that I showed at the beginning. So the idea was to actually write 3 papers for the same issue of a journal, of the Frontiers in Physiology journal. And the first paper of that series was on isometric contractions and the energy of that, right? So our energy is actually split in a few different parts. One is the volumetric part, the base material part. We have the passive part for the muscle and the active part for the muscle, right? And in these simulations, we mostly run cubes of muscle. Imagine like you have a piece of beef, and you cut like a parallelepiped, right? And then you test on that. And it's actually very common. It sounds, for a mathematician, it sounds too simplistic, but that is actually very common in engineering too. Like when they test the elastic properties, sometimes they do it with a very simple geometry. They don't go like too complicated on the geometry, all right? And so we wanted to do that. And one thing was that we wanted to study was how something called the pination angle would change the distribution of the energy, okay? So what we are doing here in these two figures here is to simply take this block, and then in the first example here with this, there is beta equals 0. We assume that all the fibers are running from -x to +x, all of them. They're all in parallel. And in this one, the only thing that changes is that we change the orientation. So now the 30-degree means that now you have a 30-degree with respect to the x-axis, all right? And then we did exactly the same test. We activated, okay? So we use this function a of x comma t to see what it would happen. And when you see the deformation of it, it's different. So we wanted to see how different it was in terms of energy as well. And here you can see that, for example, the most notorious ones are the U active, so the energy due to active forces, and the volumetric. All right. But then when you change the orientation of the fibers, then you see that the base material, the contribution from the base material is much higher than before. Okay. Which was something really surprising because we thought that maybe the volumetric would actually go up as well, trying to maintain the volume of the or nearly constant volume of the muscle. The other things that we wanted to see there was the stress and the forces, and one particular one that is the force. So in biomechanics, when they want to estimate the force of a muscle, what they do is to compute something that is called the cross-sectional area, which is essentially, you can think, imagine in this example that I have here, I have all my fibers in the x direction, right? And I compute an orthogonal plane to the direction of those fibers. And then I intersect it with the muscle, and that's my area. And so for these examples here that I show here, at muscle length 1, we should have gotten 20. That was the estimation that we got a priori from this kind of idea. And with the model, we actually got 19.06, which is quite close, I think. This is the MRI scan that I was telling you guys about. So here we subsample this data here. Why we subsample? Just because it was too much data, like the mesh was too large. So the file was too big to actually handle it within human time, because the code is not parallelized yet. So we wanted to compare. So essentially what this is giving you here is the regions where it's bulging in and bulging out, and the white is where more or less it stayed the same, all right? And so you can see, so this is the front view of the MRI, front view of the finite element code, and the back view of the same muscle and the back view. So you can see that the distribution of the frequency on the number of elements that have the same color, you could say, is about the same, not completely the same, but there are many things to consider here, right? Like we subsample the mesh, right? So that's one thing. We also had to add extra elements for this sheet of tissue called aponeurosis, okay? So those two things are kind of changed the game a little bit, but it's nice to see that the overall behavior is about the same. And this was actually also part of the first paper that I was telling you about before. In a second paper, we wanted to study how the system inside behaves when you compress it, okay? So actually, David Ryan in a paper in the 2019, he wrote, he tested human muscle, alive people, of course, human muscle, and then he compressed it and saw that as you increase the compression, right, or how do I say? Oh, as you change the penetration angle, and you would have different values in the pressure, right? So they were measuring the pressure, and they would get different numbers, and they change as they change this penetration angle that I was telling you about. This is something that we got with the finite element code, which is actually very close to what they got in their paper. I didn't put a figure here from that paper, I should have. But we got very similar results to that. Of course, we don't get the same numbers because like the experiments is plus minus error. For us, we have many approximations in the middle. For dynamic contractions, I think I'm over time now. How long do we have? 3 minutes, right? Yeah, okay.
Yeah, just about.
Okay. So in 2018, for a SICB, a Society of Integrative and Comparative Biology, we wanted to submit a paper for special issue on muscle modeling. And in there, we wanted to compare how the specific work and power of a dynamic model based on the of a 1D model, based on hill type muscle model that I described before, would compare to a 3D muscle model and to some data as well. And what we found out, so actually the actual run was that you would take your model, either 1D or 3, and you would couple it with a spring mass system, okay? And then the forces on that side will be driven by this spring mass system. And essentially, well, we took the same parameters, and we found out that dimensionality is one big effect, does have a big effect in the output. So you can see here, for example, for the power that I'm comparing here, the 1D model gets things that are order 20, up to 20 watts per kilogram. That's specific to mass. But for our 3D model, we never pass 7. So this is why we wanted to study some energy later on, because we thought like maybe the same amount of energy is being used differently by the different aspects or parts of our model, right? Ongoing work is actually on the dynamic side. So we are using the dynamic model to compare with real data. So my collaborator is not showing here, Stephanie Ross, she went on to Harvard last year and did some experiments with rat muscle. And we wanted to compare that with our model and see what happened. Actually, I don't have that here, but we also wanted to study how the penetration angle will change the specific work. And it's actually a very important key. And why I'm talking about this penetration angle all the time is because physiologists see that muscles are never in parallel and never have like fibers in parallel. They're always pinnate. So always the muscle fibers will go like this instead of going like this within our body. So here we use this activation function that is based on this excitation for these runs. So then it's essentially based on a model proposed by Sajak in the late 80s. And this is the A function that we put in to the code. And then we get this kind of results from it, okay? I'm not gonna comment too much on that. Maybe you can ask me later, but I think I'm gonna stop here. Thank you. Sorry about the delay.
No, that's no problem. Great. Thank you very much, Sebastian, for a nice talk on a very interesting application. I think maybe we have time for one question if somebody would like to ask one at this point.